So we're now on the books.
Okay, well, a few of my people might still latecomers a little bit, but most of you know I'm Tom Heitzman, the president of the Serenoa chapter of the Florida Native Plant Society. I just want to welcome everybody this evening to our February 2022 meeting already. It's the year's flying by. It's been a busy year, it seems, so far, and everything's going pretty good. We're getting some new interest on the board of directors, which is nice. So we're looking at expanding the involvement of different things, grants primarily. I think we might have somebody that's interested in helping review grants, which is for those of you who aren't aware, our chapter gives out $500 grants to schools and public institutions, not private, but we've given out quite a few $1,000s to different projects in the Sarasota-Manatee area. So that's been a really positive influence on what we're doing here. So our membership's up. Elliot Prout's taken over membership, and we have the third highest membership in the state. Miami-Dade is first, I believe. Pinellas is second, and then the Sarasota-Manatee chapter is the third highest members. So that's kind of cool. And Elliot wants to get us closer to the top even than what we are. So we'll see if we can make that happen. The field trip is this Saturday out at Lefis Key. I think the registration is full. We limited it. We're possibly going to try to start doing field trips where we don't have to limit the amount of people that are there. So we're working on that, but I think the registration is full right now. So we do have a waiting list. So if anybody's interested in attending that field trip, they can contact Susie on that, Susie Venters. And in April,
not me. I'm sorry. It would be Rachel. Still does
Rachel.
that.
Okay.
Yeah. Sorry. Yeah.
So Rachel, if you're still just check our the newsletter, and you can get any information from the recent newsletter. So the plant sale is coming up in April 15th. So that's our big fundraiser. So be aware for that. We'll be looking for volunteers, and you'll see some emails and stuff coming out for that. So that's really all I have. I think we can get right into our meeting unless anybody has any announcements from the board or anything, but we might as well go ahead and get started. So we're fortunate enough tonight to have Dr. Brad Oberle. He's associate professor of biology and environmental studies at New College of Florida. He's an environmental biologist and liberal arts educator dedicated to promoting sustainability through strong community partnerships, experience with research projects, management, staff, supervision, and communications for grants totaling over $1 million. And he's had over 20 peer-reviewed publications. So Brad, I'm going to turn it over to you. You can maybe expand on that a little bit, but take it away.
Sure. Thanks.
One more thing, just if everybody, it looks like everybody's muted, but everybody can keep their Zoom muted until after the presentation. If you have questions throughout, you can put them into the chat, and then we'll read those questions after Brad's presentation, so he's not interrupted during the talk. Okay, Brad.
Cool. Thanks, Tom. I really appreciate it. And it was nice for you to read that blurb about me, which I wrote about myself. I think it's on my LinkedIn profile. And honestly, seeing the people here reinforces how important all of those issues are to me and how impactful I hope that work has been. And that's the work that I'm going to focus on presenting. So it's great to see a lot of familiar people who I've been able to work with on this project and other projects over the course of the past couple of years. And it's great to see a lot of people who I never met and kind of regret not taking that opportunity while I was in Sarasota for a while. But I'm hoping to be able to stay involved and support some of the good things that are going on. So it's nice. Thank you very much, everyone, for the invitation. And I'm really looking forward to sharing the results of what I really personally consider to be the most impactful research project and community outreach project that I did while I was a professor at New College of Florida. And I still am for a few months. So let's see how much more good work we can do together. With that, I'm going to start to share my screen. And I'm going to take just a quick moment as I do so to sort of reorient myself to what my view of Zoom looks like. So I might lose my ability to see everybody. If that's the case, too bad. I'll just have to look at my slides for a little while. And I'm hoping that I leave plenty of time for us to have a conversation afterwards, because I think that the not only like the process of doing this work, but the products of doing this work are specifically relevant to some of the people who are participating and not quite wrapped up yet. And for that reason, your feedback is still really valuable to me at this point, because I can still include it in the papers that I'm writing and the reports that I'll be putting together for the Environmental Protection Agency. So anyways, quick summary on the summary of my introduction. It's really wonderful to see you all. Thanks for this introduction. I look forward to talking about this project, and I'm going to spend a little time babbling on, but I'm hoping to have more of a conversation afterwards. So with no further ado, I will share my screen, and fortunately I can see folks a little bit, and hopefully what you can see is my title slide, which shows a couple of images of one plant and some special people on Tidy Island. And for those of you who aren't familiar with Tidy Island, I will get people oriented in a couple of slides, but I imagine that most people are familiar with the plants that we're looking at here. So in the foreground, we can see an Australian pine, and that plant's name is a bit of a misnomer for reasons that I'll explain in a little bit. But left and right, you can basically see before and after. This is a story of Tidy Island, the place that I'll be describing. So to the left, we have one plant with 14 stems that are above 7 centimeters at 1 meter and 40 centimeters off the ground. So just kind of a location where foresters decide that a plant is a tree. So this particular plant is impressive. And so are the students who climbed up there. Former thesis student, New College of Florida, a current thesis student, New College of Florida, and some incredible field technicians. So they helped me measure these plants, part of the great team that has been able to execute this work. And to the right, in the aftermath of that plant most being chopped down, you can see my wife, Darcy. So she's hanging out back there, holding up the 0 in contrast to the 14 that the students are hanging out. That tree at this point has no stems. And Darcy is super pregnant. This was like 2 weeks before her daughter was born. And I managed to cart her out to Tidy Island just to see some of the work that we were doing. So this before and after image, for me at least, is a pretty significant representation of some of the work that we've done. But it doesn't include all of the motivation. It certainly doesn't include all the people. And I want to take a moment to acknowledge some of those people, including at least one person who's participating in this call right now. So this endeavor was very much a team effort. And I'd like to acknowledge people both at New College of Florida and at the Sarasota Bay Estuary Program who made it happen. So my colleagues in the biology department, Drs. Jane Gardner and Sandra Gilchrist, contributed tons of invaluable expertise that's really complementary to mine, and some really valuable perspectives on trying to broaden the focus and impact of my work from plants to both fish and communities. And then, of course, Julie Morris, who I'm so gratified to see on the call right now, who has really deep experience at Tidy Island and sort of brought it to my attention and some of the opportunities there. And so I'm really grateful to Julie and excited to share some of our preliminary results, which I don't think Julie has actually seen yet, because almost no one has. These are like hot off the presses as of just a couple of weeks ago. And then also, this work involved organizations beyond New College. As many of you know, my wife Darcy worked for the Sarasota Bay Estuary Program for a long time. And her colleague there, Dr. Jay Leveroni, was absolutely instrumental in getting a lot of this work off the ground, even though he's in the water there. So these are some of the most instrumental people who were involved. But then there were a lot of supporting organizations and other people who participated along the way. So most of the research that I'll be describing was funded by the Gulf of Mexico program, which is a program through the Environmental Protection Agency. In addition to that, we had some great funding through the Cross College Alliance that paid for student interns to kind of expand the scope of our work. And we just had such incredible buy-in from the community. The local community at New College, where over 30 students participated in this research. And it wasn't just New College. We also had students from Ringling College of Art and Design and USF Sarasota Manatee who participated in this work. And at least 2 or 3 people made it out to Tidy Island, got their shovels dirty, and helped sort of establish what I think will be the future of the vegetation there. So if you did participate in this work, and I haven't named you like I named Julie, that's not to minimize your contribution. Thank you. And I want to recognize that this was really a team effort right at the outset before I dig into even sort of what inspired the work or what we tried to accomplish. Very good. Okay. So I'm going to talk about the motivation, what we did, and what we found from this particular research project that happened, maybe 12 miles up the road from where we would normally be meeting at Selby Gardens. So the motivation. Mangrove can be a little bit of a confusing word because it has 2 meanings. It can either refer to a plant or to a habitat. When you're talking about a mangrove plant, you're talking about a plant that basically is adapted to living in salt water. And when you're talking about a mangrove habitat, you're talking about the kind of habitat where you see mangrove plants actually growing. So to bring it a little bit further, these specific habitats have conditions that make it really hard for plants to grow because they're almost always salty. Plants have to have specific adaptations to deal with salt. And these are things that are really hard for plants to do. So the plant on the left is a black mangrove, and people in the Native Plant Society probably almost uniformly know the best and funnest way to identify this plant is to lick one of its leaves, right? It's a salt excreter, which is a pretty or extraordinary thing for any plant to do, to let salt water go all the way up through it. When it gets to the leaves and photosynthesis and transpiration happens, the salt gets stuck, right? So it actually forms crystals on the back of the leaves, and the whole anatomy and physiology of making that work is really extraordinary. But not every mangrove does it the same way. The one on the right is a red mangrove. And if you were to cut the roots open and do some fancy staining, what you would basically find is a incredibly advanced cork filter that excludes the salt from this plant's vascular system so that it only has fresh water flowing inside of it. And it's pretty impressive. That biological system has actually inspired some engineering approaches to desalinating seawater. We could go on and on about the plants and their amazing adaptations, because there are actually even more difficult challenges for plants to grow in mangrove habitats. Because they are regularly inundated, these plants have aerial roots that basically help them oxygenate the root system so roots can do what they can do, and that is grow and get nutrients. And because they're growing in these coastal areas, they do get a little bit of wave energy. And for that reason, mangroves are built to be tough. They have really dense wood, and the branching structure kind of dissipates wave energy when it comes in and out. So what we're talking about here when we're talking about mangroves is both a plant and a habitat that are really closely tied to one another through some pretty impressive adaptations. Not every plant can make this happen. Mangroves, both evolutionarily speaking and geographically speaking, are pretty rare. So Florida only has 4 species, and that's a little generous because honestly, I wouldn't exactly consider buttonwood to be a species of mangrove. Even so, there are only a couple of other species that you find in the Western Hemisphere. And then even when you go to the Old World, where mangrove forests are much more diverse, there's fewer than 60 species of trees that can actually grow in this habitat. So they are pretty remarkable plants. And geographically, they don't actually occupy a lot of territory, right? They only grow in the tropics, they only grow along the coast, and they can't grow in places that have too many tides or too many strong waves. So they wind up representing globally about 0.5% of all the coasts worldwide, right? And an even smaller proportion of all the forests. So you might think with few species and not much of a footprint that these plants really wouldn't be worth focusing on, but they have a disproportionate impact. So if you find little particles of carbon floating around in the ocean, 1 out of 10 came from a mangrove. And when you look at all the coasts and how much carbon they store, which is really important if you're familiar with the term blue carbon, well, mangroves contribute about 15% of all the blue carbon storage. And if you compare them to tropical forests around the planet, mangroves store 30% of all the carbon that tropical forests actually store. So whether you're talking about the plant or the habitat, they might seem so rare as to be insignificant, but they have a very disproportionate impact on really how carbon moves around the planet. And that's not the limit, right? Carbon is life, right? It's what we're all built out of. And as these plants grow, they feed really diverse and economically important food webs. So this little infographic is just one illustration, right? Mangroves support incredibly highly productive food webs, and they feed more than 210 million people every year, right? So whether you're interested in the plants, you're concerned about the climate, like if you want to eat well, right? Understanding how mangroves work and making them work better should be a big priority. They are an absolutely vital food source. So given that, the importance of mangroves, you can find mangroves growing all over Sarasota, and people who go to Lefis Key will see a really great example of a restored mangrove forest. They are kind of the dominant vegetation along the shoreline. And even though they are fairly prevalent today, they are still under threat, right? And so I'm going to talk about a very important threatened mangrove in Sarasota Bay called Tidy Island, right? It is a partially developed peninsula that's in the north end of Sarasota Bay in Manatee County. And about 2/3 of that island, 188 acres, are part of a research reserve that was deeded to College of Florida to do teaching and to try to protect the environment. And I just told you it's called Tidy Island, and I just told you it's a peninsula, right? You might have caught a little bit of a cognitive dissonance there. Oh, what's going on? What do I mean when I'm talking about this peninsula called Tidy Island? Well, the history here is actually kind of interesting because it illustrates part of what mangroves do. So if you look at the very earliest map of Sarasota Bay, this one was generated, as the image says, in 1883. Tidy Island was in fact an island, in the sense that there was open water separating exposed dry land from the rest of the shoreline in Manatee County. And the tidy here, anybody who's actually spent time there, doesn't refer to it being like neat or clean. It's probably a little bit of pigeon speak for tidal, okay? So it was a tidal island in the 19th century, right? Where at high tide, there was open water that was separating a little upland area from the rest of Manatee County. But from this point forward, things really started changing at Tidy Island. And honestly, what probably changed them was the mangroves themselves. So if we look at the first aerial photograph of Tidy Island from the 1940s, you can still see that upland area in the bottom of the image, right? The top of the image, the dark gray, that's all mangrove swamp. And now you can probably try to imagine paddling some kind of a boat between the north side of Tidy Island and the south side of Tidy Island over there by the rest of Manatee County. It's not going to happen, right? And the reason why it's not going to happen is that the mangroves themselves actually build land, right? They're so productive, they store so much carbon, they trap so much sediment that it actually causes land area to accrete. And in this case, the mangroves establishing at Tidy Island probably glued the island back onto the mainland of Manatee County. And that's an interesting biological phenomenon. It turns out to be kind of interesting from a legal perspective, because Florida law is written in such a way that accreting land, depending on which direction it accretes, belongs to different property owners. Anyways, not really my area of expertise. Like my area of expertise, obviously, is the plants and what they're doing around with the environment. But at this point in time, Sarasota and Tidy Island were still pretty sleepy places, right? So there were a couple of families of homesteaders that were living in the upland area of Tidy Island that's been above water for, probably a couple of thousand years. They would probably run cattle out there at low tide and then bring them back to Manatee County a little bit later on. So this is a pretty good representation of what life was like on Tidy Island once the mangroves started growing back in. But like a lot of things around this part of Florida, big changes occurred in the 1960s and 1970s. So this is another aerial photograph where you can probably see this quilt work, right? And then some deep scars. And often when I show this picture and describe these changes to Tidy Island, I have to like roll up my sleeve, which I can't really do yet. I mean, these are actual scars. It's useful to think of it this way. So what we can see in this dramatic habitat alteration are the effects of two kinds of dredging. So to get people to live here, they want to be able to take their boats around, and to take their boats around, they need deep water channels. So in the northern border of Tidy Island, where you see the dark gray, that's actually deep water, and it's all deep water that has been dredged out. And then this quilt work that occurs throughout the mangrove swamps, these are mosquito ditches, right? So this was an attempt, kind of ineffective, of controlling the nuisance mosquito populations that live in this swamp like all other swamps. The idea is you make these channels, gives fish access, fish eat mosquito larvae. That's a natural way of controlling mosquito populations. But both of these changes to make coastal property more valuable, take your boat, don't have to deal with swarms of mosquitoes, had really big impacts on the swamp, right? All that spoil has to go somewhere. And when you pile it up, you displace mangroves and you create this new habitat that winds up potentially attracting other kinds of plants and maybe other kinds of plants that we don't like so much. And so what you can see in this image is a contemporary digital elevation model. And basically it's just something that sewage departments use to figure out which way poo flows, right? So it's really detailed. And here, how white a part of the map is how high it is above mean high tide. So you can see these darker channels, and you can see these bright white spoil ridges that follow the navigation channels that are pretty much all around Tidy Island. And then of course in the far south, you can see the built up areas that are part of the condo association, the Tidy Island Condo Association. Now is probably a pretty good time just to let folks know. I'm talking about Tidy Island and the fact that it's owned by New College as a mangrove preserve, but that does not apply to the private property that's down here at the bottom that owned by the Tidy Island Condo Association, and they appreciate their privacy. So please don't go up to their gatehouse and tell them Brad Oberle sent you. See if you can go wandering around in the swamp. It doesn't exactly work that way. If you're really interested in going, we can have a conversation. There are some other ways of getting there, but for the most part now it kind of takes a boat. Okay, disclaimer aside, this is kind of the situation at Tidy Island. Okay, so it was an island at one point, mangroves grow, they glue the island onto the mainland, and as they're doing all this great earth building work, people come in and start moving things around, right? They're dredging channels to get boats here and there. They're dredging other channels to try to bring fish in to eat all these mosquito larvae. And the consequences are a really transformed landscape. So these two forms of major disturbance, navigation channels and mosquito ditches, really alter the hydrology in both the terrestrial and the marine communities. So plants and fish really have to respond to these changes that people made. And the changes invited some of the least desirable species of trees that we find in this part of Florida. So I referred to one of them before, Australian pine. The pines here is in scare quotes because it is definitely not a conifer, right? From a botanical standpoint, these plants are much more closely related to oaks. The Australian part is accurate, and I actually saw some of these plants relatives growing in Australia, pretty scraggly over there. And Australians refer to them as she oaks, which is kind of interesting. And that is a reference to the quality of their wood, which is actually quite a bit like the wood of some species of oaks. So this seems all good. Plant that grows fast, it can grow in coastal situations. That's part of the reason why it was introduced. The issue is it grows tall and it avoids salt water by having very shallow roots. So when we get a bad storm like Ian or Irma or any other tropical system, these things have a very high risk of falling over and really causing some devastating damage. So some of you at least probably remember the Australian pines that used to line the road out to Lido Key, right? Those came down during Hurricane Irma. And if the storm had been worse than it had proven to be, that could have really stranded people on those keys, and it could have been a major issue, right? So one example of a plant that exploits the kind of disturbance that we saw at Tidy Island, and it can compound problems for people in our community because they just don't perform well during storms. And this is just one of the new plants that exploited the disturbance at Tidy Island. Another one, scare quotes again, Brazilian pepper, not a pepper at all, right? It's not in the pepper family. It's not in either pepper family, whether we're talking about chili peppers or black pepper, right? It's actually in the poison ivy family, right? The Anacardiaceae. And like a lot of plants in that family, it has some pretty nasty toxins that occur in some of its tissues. I personally have never had an issue with the fruit or the leaves, fortunately, because I've handled a lot of them. But there was one time I was collecting a soil sample and brushed my arm past a cut root and wound up with a nasty chemical burn that stuck around for 2 months, right? So this plant is protected chemically, and people who are more sensitive than I am have a lot of issues with these particular species. So there are reasons why we would potentially not want to keep these plants around. And partly it's just self-interest. These plants cause harm, but also the mangroves themselves are so important. We really don't know if the presence of these plants in the mangrove habitat harms or helps the many incredible things that this particular forest type does. And so a big question, and it's true for New College, it's true for Manatee County, it's true all over Florida, is does removing these invasive species affect the services that mangroves provide? And the services I'm talking about here are things like storing carbon, filtering water, and feeding fish, right? If we can get these plants out of these habitats, do mangroves actually do a better job of providing those services that we rely on all the time? And that's precisely the question that we sought to address with a cooperative agreement with the Environmental Protection Agency that I mentioned right at the top. And the idea here is that we have this natural habitat that works in a particular way to provide all of these valuable services, right? And our question here is, can we remove invasive trees from Tidy Island in such a way to increase the amount of productivity in the habitat, how fast the plants grow, how many fish they feed, and the value that it provides to our communities, specifically in terms of storing more carbon and feeding more fish. So it was an opportunity to actually do some pretty practical research at Tidy Island and hopefully make the habitat better for the people not only who live right next door, but all the people who live around it in Sarasota Bay. And the way that we did this was by putting together an experiment where we really carefully measured what was happening at Tidy Island the way that it had been for the past 80 years, right? Totally overrun by invasive plants. And then we changed things up. We did experimental ecological restoration to see how different methods of controlling these invasive plants actually affected the habitat and what happens in the habitat. So now that I've given you the goals of this project, I want to talk a little bit about what we did. And this is a more contemporary map of the area where we focused our efforts. And all the areas that are colored here are areas where we have different invasive removal treatments. In some places, we just take the plant, we girdle it, and let it die standing exactly where it is. This is what I'm going to refer to as kill in place. In other places, we leave the invasive vegetation exactly the way that it is. This is what I'll call a control. And then we have two different treatments here that actually involve chipping up these big invasive trees and moving their biomass around. So we have a plus mulch treatment and a minus mulch treatment. The mulch here isn't mulch that we're bringing onto the site. It's the mulch that we are creating out of the invasive vegetation. So the minus mulch treatment here, we have our invasive plants, we kill them, we chip them, and we actually physically raked all of the mulch off of those sites. So this kind of simulates some invasive tree control that happens in coastal Florida, where you either cut down the trees and truck off their biomass, or you cut them down, you actually burn them, right? So this is kind of the most extreme treatment that people use for these particular invasive species of plants. Plus mulch was honestly just kind of a convenience thing. You just raked up a bunch of mulch, you could dump it on another plot, and that way, if there wasn't a lot, we could actually double it up to see if there was actually an effect. Okay, so these are some of the treatments that we're looking at. And this map gives you a sense of where we were implementing them. And it's not all over Tidy Island. It's just on some really high elevation areas where the invasive trees had a very strong foothold. Okay, so that is our main focus. Our main focus is on these areas where we had lots and lots of invasive plants. And our monitoring focuses on this area that's in basically the northeast corner of Tidy Island. It's a really large spoil area, probably got that way because it's right next to the community at Coral Shores, and a lot of dredging happened there. And so a lot of that dredge spoil showed up on this particular part of the island. On this part of the island, we distributed a set of 24 monitoring plots. These plots are pretty small. They're only about 100 square meters, right? So 8 meters on one axis, 12.5 meters on the other axis. They're all centered at mean high tide. And the idea here is that we can look at what's happening in trees, litter, and soils in both the mangrove habitats and in the spoil habitats where the invasive plants are growing. And we've got some replication built into this so that we're not just looking at one place where we did one thing to the invasive plants and comparing it to another place where we did another thing to the invasive plants. So this is kind of how we set up our monitoring, both before and after the restoration that we were attempting to do. So we're looking at what's happening in these spoil mounds with the invasive plants. We're looking at what's happening in the mangrove forest that's right next to our invasive plants. And the other thing that we're doing is we're actually looking at what's happening in the channels that were dredged out to actually make all of this disturbance happen in the first place. So there are two little blue boxes I just highlighted. These are two ditches. In these ditches, we have a kind of fish-focused counterpart to our experiment on land. So we're also doing monitoring here. We're monitoring both water quality and fish communities. And we're also doing an experiment here. Some of the mulch that came from our invasive plants in our focal area, we're actually bagging it up, sticking it in one of these ditches to see how the fish respond to an extra dose of invasive plants. So for this work, I definitely want to credit my partner, Dr. Jean Gardner, because I don't know how to measure fish, and she is an expert. So it's part of what made our interdisciplinary research both really rewarding and really exciting. So I hate to bore a bunch of native plant people with a little fish stuff. I've got one picture of a fish a little bit later on. Hopefully it doesn't affect people too much, but I will be talking a little bit about the marine focus aspect of our experiment as I move along. And the work that we're doing here is really big scale, right? And it requires some really heavy equipment, and it requires some equipment and other kinds of modifications to parts of Florida that we all share, right? So we all share the water in Florida. And for this reason, it was really important for our team to make sure that we were doing it in an environmentally responsible way. And we worked with both the Army Corps of Engineers and the Southwest Florida Water Management District to make sure that our experimental approach was consistent with their organizational responsibilities to protect the environment for all the people in Florida and around the country. So we had some great partners at some environmental organizations, environmental consulting firms, to make sure that we crossed all those T's and dotted all those I's. So actually, before I get into results too deeply, I actually would like to pause here because I realize I'm just blazing through my talk, and that's a good thing. But I want to open up the floor for any questions that people have about mangroves, about Tidy Island, or about what the goals were for our experiment before I start digging into the kind of nerdy scientific results. So I'm not getting any takers. Maybe you all are mesmerized by this picture of Derek Harvey, wondering what in the world he's doing, staring at a piece of wood floating in the air. So maybe I'll just cruise right along.
I do
but
have
I'll-
one question, if I may. Were you worried at all about the soil disturbance when you cut down some of these invasive trees? Did it affect the underlying layers at all?
Andy, that's an excellent question. So I mentioned these monitoring plots that we set up, and because I'm a plant person, it would be a pretty smart assumption that we were only monitoring the plants, but in fact, we were also monitoring what was happening in the litter and in the soils. And by monitoring it, that might be a bit of an exaggeration. We just measured it a couple of times, right? So we measured all the plants before we got started. I'm going to present some analysis of those results here in a moment. But we also measured how much litter was on the ground. And I say litter here, I'm not talking about like beer cans and, like PFDs, although there are unfortunately plenty of those around Tidy Island. What I'm talking about here are leaves, right, and twigs. I'm talking about plant litter that's built up. And then the other part that we were monitoring, as I said a second ago, is we were monitoring the soils. So we were monitoring the density of the soils and the carbon content of the soils. And the goal for all this was to basically figure out, Andy, exactly what you were asking about, right? Do these restoration activities not only influence the plants, like the invasive plants, which are the target, right? Do they influence the growth of the mangrove plants right next to them? Do they influence the kind of litter that's built up on the ground, whether it's mulch or leaf litter or something else? And then what impacts does that have on the soil underneath them? And my focus is being somebody who's very interested in climate science and the carbon cycle, was figuring out how much carbon was stored in all those layers. But we can figure out generally, if there were impacts in those places. So the idea is that we went through, measured them all to begin with, and then we do our invasive treatments as an experiment, like replicated with different levels and a control and everything. And we can really measure like, yeah, okay, so if you run a mulcher in there, you're going to lose this much carbon from the soil. And we'll get to those results in a little while. So excellent question. That was something we were concerned about and concerned enough to include it in our measurements. And through those measurements, get a really comprehensive picture of what was changing at Tidy Island as we were doing this experimental restoration project. Okay.
How long do you project to monitor? Like for another year or 5 years?
That's an awesome question. We are out of money. Our grant terminates this month, and so we don't have any additional funding for any future monitoring. Now, having said that, the plots that I put up are set up. And so anybody who has access and skills could remeasure what we originally measured relatively easily. And so it wouldn't take a lot of money and it wouldn't take a lot of time to go back out to Tidy Island to do this again. But it would take a little bit of money and a little bit of time. And so I would be interested, kind of depending on what my next employment situation looks like, in continuing some of this work. And I'll also mention that we did some other really cool stuff at Tidy Island while we were out there to do some experimental revegetation. As a matter of fact, Tom, you help with that work because you help supply some of the plants for it, right? Those are some sort of ongoing projects that would be really cool to keep track of. And I'll describe a little bit more of what's happening there. But the bottom line answer is we're pretty much done with monitoring. It was kind of a before and after situation, but we're set up to continue looking at least what's happening to the plants, the litter and the soils for anybody who's got the access. Any other kind of questions up to this point, just about how we, what we were trying to do and how we got things set up?
There's a couple in the chat.
Oh, hi, Karen.
Hey. I see Elliot Prout asks, is the developed area of Tidy Island a natural upland or dredge spoil from the Gulf Intracoastal Waterway?
Elliot, that's a really cool question. And honestly, Julie is the expert because Julie and Jono helped do some of the initial vegetation surveys on that part of Island before it was developed. So crediting them at the outset, I'll try to summarize what I learned from them about the history of the vegetation on that part of the island. It was almost certainly a natural upland. And one of the things that supports that is not just the vegetation they found growing there, but also some of the archaeological evidence on the site, right? So there are, there's evidence of indigenous people who lived there for probably a solid couple thousand years, right? And so the fact that they were there living their lives, leaving remains, indicates that, yeah, that part was probably upland for a while. And I think the topography is part of the reason why that area attracted the development, right? And there were some, what we understand, kind of crazy proposals to develop the swamp part of Tidy Island, but they all fell through just because it was a little bit too technically challenging to do. And that's fortunate for everybody, because nasty as that swamp is, it's providing us all with a lot of services. So yeah, Elliot, great question. That part is not dredge spoil. And then that way, this part of Tidy Island is different from some of the other islands around Sarasota Bay, like Sister Keys, for example, where a lot of that was dredge spoil from the intercoastal waterway.
There's a couple other questions, but I think we can
Yeah.
wait till the end of the talk and I can read some of those in the chat.
Awesome. Tom, I will defer to your MC expertise. Thanks. And thanks for the questions. I'll try and hopefully break things up a little bit as I go along, but I will briefly introduce you to Derek. Derek is one of the 30 or so students who was involved in the research at Tidy Island. And what Derek is helping us do is weigh a carrotwood tree. And the reason why Derek is doing this is that people don't actually know a ton about carrotwood. I didn't even introduce it to begin with. Carrotwood is kind of our local invasive tree. And it's local in the sense that it was actually a Sarasota County IFAS extension agent who promoted this tree for landscaping in the 1960s. And the idea was that it would never grow in habitats where you didn't want it. And immediately it started growing everywhere, right? So this is kind of the third player in the story of the invasive plants that are growing all over Tidy Island. And simply, we just didn't know enough about it, right? And to learn more about it and to be able to measure what was happening in the vegetation out there, we basically had to break a couple eggs, right? So we carefully measured and harvested 19 carrotwoods, 15 Brazilian peppers, and 12 natives that we used for revegetation. So we measured them all really carefully. We cut them down, we weighed them. And then what we did was we built a statistical model that helps us predict how much one of these plants weighs from just a simple measurement of how broad it is, how broad the stem is, about 5 feet off the ground. And I'll spare you a detailed explanation of this graph. Part is grayed out that you don't really need to see. If you don't have really specific information about these trees, you wind up with really bad guesses about how big they are. We built this specific information in order to improve how accurately we could estimate their size. That is, like how heavy they are, how much carbon they store, from how big around the trunk is. And we actually do a really great job. I was really pleased with this kind of result. And we actually did it in a fun statistical way that I think will help researchers in places like Tidy Island, where you have lots of new trees that people don't know well, and you don't have an infinite amount of time and the confidence with the chainsaw to just continue to cut trees down all the time to build these kinds of equations. So this was really the first positive outcome of our research at Tidy Island. We built new tools for figuring out how much these invasive trees actually affect the environment, and specifically how heavy they are based on how broad their stems are. And the tools actually work pretty well. Okay, so now that we have these tools in place, we can actually measure all the trees and figure out how heavy they are and what kinds of impacts they have on the environment. So I mentioned before, we have these 24 inventory plots all around the spoil region in the northeast corner of Tidy Island. In these inventory plots, the team of Dorn here on the far left, Gus in the middle, and Simon on the right, and I measured over 600 stems in the January and February of 2020. This is how we kind of established our vegetation baseline. And this is how we got introduced to our tetradecagon Australian pine, this crazy plant that I introduced at the very beginning of my talk that resprouted and created all of these different stems. And what we found when we look at all these plots are one thing that we already knew, and another thing that was kind of interesting and surprising. Because the thing that we found that we already knew, it's an invasive plant bonanza, right? These invasive trees, the three of them that I mentioned here, Brazilian pepper, carrotwood, and Australian pine, contribute more than 95% of all the biomass in the spoil forests at Tidy Island. So if it weren't for one gorgeous live oak tree that was the very biggest plant that we had in any of our plots, right, this number would have basically been 100%. Okay, so not super surprising that there are so many invasive trees growing here. Just a glance at the habitat would tell you as much. The thing that was kind of surprising was you only find one or the other. Okay, so the graph that I'm showing here is a little bit hard to wrap your mind around, but it basically shows differences in the vegetation structure based on the distance between the points in the plot. Okay, so there's a point for every one of the plots that we measured, and there are some names that I have here for the different species of invasive trees that are around. So if all the plots had the same 3 invasive plants in them, then all of their names would fall right on top of each other. But instead, what we find are the names of these invasive plants on the points of a triangle. They're as far apart as they can get. And what this means in a kind of more tangible sense is if you're on Tidy Island and you're on the spoil, you're either standing under a canopy of Australian pines, you are battling your way through a thicket of Brazilian peppers, or you are absolutely in a prison of carrotwood saplings, right? So it's one plant or the other that kind of form this checkerboard where one is dominating and another is not. So this is actually kind of interesting from a basic science perspective, because ecologists have thought for almost a century that if you put the same plants in the same habitat, there will only wind up being one winner, right? And that in this particular habitat, all these plants are invasive species, they're all trees, they should all have pretty much the same characteristics, and they're all growing in dredge spoil, right? So it's not like you have swamp over here and upland over here, right? It's all dredge spoil. But nevertheless, we have these different communities of plants growing in different spots in this relatively homogeneous area. Okay, so that's true today. It's kind of an ecological puzzle. Why do we have these plants that is each kind of dominating its own little corner of Tidy Island? Well, it's only the case for right now. We had almost 70 seedling monitoring plots where we were basically looking to see what was happening in the next generation. And with the exception of one plant, every single seedling that we measured on Tidy Island was a carrotwood seedling, right? So if you look at the canopy today, it's kind of diverse. It's not the kind of diversity that you would like, because it's almost all one of these 3 species of invasive trees. But if you look at the future of Tidy Island, it's only carrotwood. This is the only plant that is actually recruiting out there. So our second outcome from this inventory was basically that carrotwood's taking over. And so we kind of have 2 mysteries from our first examination of what Tidy Island is like. The first one is, why are these different invasive trees stuck in different corners of Tidy Island? The second mystery here is, what is it about carrotwood that is making it so good at getting established as the future of Tidy Island, the only thing that's going to wind up growing there after all the other invasive species wind up kicking the bucket? Well, to answer this question, we dug a little bit deeper. So at each one of our inventory plots, we measured both litter and soils, and then we can analyze the relationship between the litter and soils and the rest of the vegetation to get a better understanding of really the nuts and bolts of how Tidy Island works. So I love these kinds of graphs. They can be a little bit intimidating. This graph is meant to represent the relationships between a lot of the different variables that we measured at Tidy Island. And I'm going to point you to the 3 boxes here that have plant names in italics. So casuarina is our Australian pine, Cupaniopsis is our carrotwood, and the arrows that connect these boxes show cause and effect relationships in our data, right? If that arrow is red, it means it's negative. And so the big area that you can see on the far left is one that I already talked about. There's a negative arrow that connects casuarina adult biomass to Cupaniopsis adult biomass. This means if you get more casuarina, you wind up with less Cupaniopsis. These 2 tree species are competing with each other. And this kind of explains while you either find a patch of casuarina or you find a patch of Cupaniopsis. Cool. So that's what one of these red arrows with the negative values means. The green arrows that have the positive values represent positive relationships. And so one that probably shouldn't be very surprising is that the more Cupaniopsis adult biomass you have, the more Cupaniopsis seedling cover you have, right? More adults, more seeds, more seedlings. Nothing really surprising here. What's interesting is when you include information about both litter and soils. So those of you who have been out in a forest like the forest at Tidy Island and have been underneath casuarinas have probably noticed a couple of weird things about those habitats. The first one is you basically don't find any plants in the understory unless they happen to be Cupaniopsis seedlings, right? The other thing is that you're walking on kind of a weird squishy carpet, and that weird squishy carpet is litter. That litter are the tiny photosynthetic branches of Australian pines that give them their name pines, right? The weird thing is pines needles are actual leaves, whereas Cupaniopsis needles are little tiny reduced branches. It's a different way of living your life as a plant, and it makes them have litter that's both poisonous to other plants and very difficult to break down. So for the most part, that litter is what helps casuarina be the boss. It suppresses its competition, and it makes it so that only casuarina can come back. The thing here, though, is that Cupaniopsis appears to have broken the code. And what I mean by that is that as casuarina grows, it has a positive effect on how much litter is there. Of course, same thing is true of Cupaniopsis. But when we look around the places in Tidy Island, the places that have the most Cupaniopsis seedlings actually are the places that have the deepest casuarina litter, right? So there's kind of a weird indirect effect here. Okay, what I mean by that is that if you look at adult Cupaniopsis and casuarina, that is carrotwoods and Australian pines, you don't find them in the same place at the same time. They compete with one another, but they both make litter, and the litter that casuarina makes actually makes better conditions for Cupaniopsis seedlings to grow. So even if this litter is helping these two adult plants compete with one another, right, the litter is setting the stage for Cupaniopsis to take over. There's another little box here for soil carbon and nitrogen ratios. This is also connected in a kind of more tangential way. Soil carbon and nitrogen ratios have to do with soil quality. And the more quality of the soil is, the more Cupaniopsis seedlings that we see growing there. Okay, so basically it looks like Australian pines are kind of sowing the seeds of their own demise, right? They are creating conditions on Tidy Island where Cupaniopsis seedlings actually do a better job of taking over. And to kind of put the nail in the coffin of this cause and effect relationship, we actually set up a really fun germination experiment that we did in the labs at New College. So we took seeds from our 3 nasty invasive plants. We also took slash pine seeds just to have a kind of native plant represented. And then we took litter from all these plants as well. And we did a crossed experiment where we took every kind of seed and grew it under every kind of litter. And then we also grew these seeds on bare soil, kind of what you would expect to find when these plants are first getting started. And what we found was actually pretty cool because it was consistent with what the patterns are when you measure the vegetation today. Australian pine germinates best on bare sand, and it germinates much worse underneath its own leaf litter. That's what you can see by this little green blip here in the far left. This is the seedling survival probability in our experiment for Australian pine seedlings that are growing on bare sand. If you put them under any kind of litter, they basically don't have a shot of surviving past their little seedling beginning stages, right? The heights of these bars here are much higher for carrotwood. Carrotwood seedlings do a much better job of surviving. The thing that's really interesting here is they do the best job of surviving if they are under Australian pine litter. And they do an even worse job of surviving if they are on bare sand. So Australian pine smothers its own seedlings, and it smothers plenty of other invasive plants, but it actually fosters the next generation of carrotwoods to take over. So from our baseline inventory, we basically have 3 big takeaways. the first takeaway is that we can put together new tools to understand what kinds of impacts these plants have on their environment. The second takeaway is that it's pretty much an invasive plant show with carrotwood taking over. The third takeaway is that it's not just about the plants influencing the environment, it's also about their litter, right? Their litter plays a really big role here. And this presents us with a hypothesis. The hypothesis is basically this question: will removing the invasive species along with their litter actually do the best job of restoring a little bit of diversity at Tidy Island? And this is how we did it. We paid an environmental contractor to actually barge a forestry mulcher onto Tidy Island. And I'm going to warn folks right now to turn down their mics a little bit, because I'm going to play a video and it's kind of loud, but it's also kind of awesome. I took this video just a little while before my daughter was born. It was kind of my last hurrah at Tidy Island before like 2 months of no sleep. Okay, so carrotwood's right in front of me. This is Michael operating Richards Environmental's forestry mulcher, and he is taking out an adult carrotwood tree in like 12 seconds. And I had spent a week cutting up and dissecting a tree of this size in this particular area before. I mean, it was like the 8-year-old boy that is alive in me was really, really into this part of the project. It was actually like a fun way to kind of start my journey into parenthood. But the bottom line is that we took Tidy Island from a place that was basically just a coliseum of invasive plant competition and transformed it to this, right? So this is a perspective almost from that same place where I took that video after the forestry mulcher did its job. And you can see there's still some Australian pines hanging around. Those are actually too tough for the forestry mulcher to chip up. So what the open spaces here are basically the spaces where there had been carrotwood before, or where there had been Brazilian pepper before. And this is a satellite image of Tidy Island before the forestry mulcher did its job. And this is after, which I think is pretty fun. You can actually see the results of our experiment from space, right? So we have our kind of baseline understanding of what's happening, all invasive plants growing in one habitat or the other. The litter seems to be the key to determining that it's only ever going to be invasive plants. And now we've done our experiment, right? We've got some control spots. Don't mess with those invasives. We've got kill and place spots where all the biomass stays. And then we've got places where we chipped up those invasive plants and either removed all their biomass or actually doubled up on the biomass in a couple of other spots. So here comes the fun results. We remeasured all the same aspects of a vegetation structure so we could actually calculate differences in the biomass of the trees, right? How much carbon they stored, how much carbon was in the litter, and how much carbon was in the soils. Good news first. Getting rid of these invasive plants transformed the plant biological diversity at Tidy Island. We went from basically having 2 species on the spoil, carrotwood and 1 sable palm, to having 22 species of different plants that showed up in our plots, our seedling inventory plots at Tidy Island. Of these, 15 of them were native plants. And interestingly, consistent with our analysis of how the vegetation worked before we got started, it was only the treatment where we chipped the invasive plants and removed their biomass where we saw a significant increase in the diversity of plants that were present on the site. So, as we expected, getting rid of the invasive plants and their biomass actually changed Tidy Island to be a much more welcoming place for other species of plants. So this is a really cool and gratifying result. We made a better space for native plants at Tidy Island by controlling the invasive plants and doing it in a specific way. That's the good news. The bad news is this decimated how much carbon was actually stored in these habitats. We lost 15 kilograms of carbon per meter squared from these spots where we removed the invasive plant biomass. Now it's kind of hard to connect that number of 15 kilograms per meter squared to anything that would affect the decision you'd make. So to put that in better terms, if we had removed all of the invasive plant biomass from our entire project area, we would have generated the same carbon emissions that 1,000 Americans make in a year, right? So the carbon costs of managing the invasive species at Tidy Island for native plants are considerable. But this only focuses on the spoil mound habitat. And even though the spoil mound habitat is important, it's actually just kind of a small fraction of what's happening at Tidy Island. Do we get some kind of a boost from the rest of the habitat? We do. So mangrove trees that were growing next to the invasive plants actually grew faster after we removed them. They added about an additional 2 kilograms of carbon compared to the mangrove plants that were just continued to grow next to those invasive species. So the invasive species suppress other invasive species growth. And we have some strong evidence here that they actually suppress mangrove tree growth as well. And because this was only about a year later, there's a chance that stimulated mangrove growth will actually kind of offset some of the carbon that we lost when we removed the invasive tree biomass. But unfortunately, it's not this simple. The reason why is our mangrove soils actually lost carbon when those soils were next to places where we removed the invasive plants. So in the mangrove habitat, it was kind of a wash. The trees grew more where we removed invasive species, but the soil lost more carbon, and these two effects cancel out. So we can't count on the mangroves to replace the carbon that we lose when we control the invasive trees at this habitat. So this is one perspective on what's happening in the broader ecosystem when we do our invasive plant control. But remember, we were also looking at water quality and fish habitat. So I'd like to just quickly present the results of some of our monitoring efforts here. One thing that was cool is when we got rid of invasive trees, fish abundance increased in our control plot. But fish abundance decreased where we added invasive plant mulch. So invasive plants don't like invasive plant mulch. Native plants don't like invasive plant mulch. And fish don't like invasive plant mulch. This stuff is pretty nasty in the ecosystem, and it doesn't seem to be doing any kinds of organisms any big favors. We also found that overall, the number of different fish species decreased after our restoration, and it decreased significantly in the ditches where we added invasive plant mulch. So again, there's really good evidence that we have for the entire ecosystem that you have to be careful with what you are doing with this invasive plant mulch after you treat the trees that are growing on one of these sites. And fortunately, we were able to work with people to try to make sure that the future of Tidy Island looks better than the past by not only letting some native plants naturally grow back into the habitat, but actually putting in some native plants that are more interesting to give them a little bit of a head start. So with 50 plus people in the community over a really hot weekend a couple of Septembers ago, we actually managed to plant over 2,000 native plants. And this, my hope is, will result in a future for Tidy Island that looks better, looks more diverse, and actually does store more carbon. And I'll also point out that we've been doing similar things for the fish community there. We've been doing some experimental stock enhancement using snook, which we hope not only compensates for some of the fish that may have been negatively impacted by our restoration, but also from some of the other changes that have occurred, like red tide, while this project was actually going on. And our impacts in the community through this project aren't just limited to the landscape at Tidy Island and the people who have worked there. We have also worked with lots of middle and high school students from around the Sarasota Manatee community to help them understand how the health of the bay affects the health of its citizens. So we had 75 students who conducted their own research on invasive plant mulch and water quality. And we're actually able to measure that the students who participated in this process actually learned more about the carbon cycle. They could describe some things that actually some of our new college biology students have a hard time understanding. So our project here, I think, had a lot of positive impacts, from basic science to habitat management to what folks know in the community. And this might seem like a kind of unceremonious end, but thanks. It's been really wonderful to be a part of the community and share some of the results of my research. And I'm a little bit over, but I still have some time, and I'm happy to answer questions that anybody happens to have if they have got some time to share them with me.
That was great, Brad. Very nice talk.
Mhm.
It does bring up a lot of questions. So if anybody wants to take them from the floor, and then I'll go ahead and read some from the chat room.
I have a question. You talked about the when the mangroves grew faster
Mhm.
after you removed the invasives, but then the soil carbon negated that. Long term, do you think that balance might change?
Karen, that's such a great question. And I think it's possible. My guess as to why we saw that effect was actually some of the litter from the invasive plants makes it into the mangrove habitat. And researchers at other places of Florida have done experiments to show that at least Brazilian pepper litter actually keeps mangroves from growing very well. So I think that explains why we saw the sort of bounce back of tree growth after we removed the invasive plants. And I think it also probably explains why we saw the reduction in how much soil carbon was present, because that litter, even if it keeps the mangroves from growing, it adds carbon to the soil as it decomposes, right? And the question I think that's really pivotal that you point out is, will mangroves without those invasive plants present start rebuilding the carbon that we see that was lost in this kind of transitional period right after we kill those invasive plants? I think it's possible. We just haven't actually seen it yet. And I think that would be a really cool thing. And considering that most of the carbon that shows up in mangrove ecosystems is stored in the soil, it's really in the soil where we need to start seeing some differences. Excellent question. Thanks for that, Karen.
One from the chat, you mentioned how mangroves feed like 231 million people. Is that primarily through the fisheries they create, or the obviously mangoes aren't edible to people?
Yeah. So it is mostly through fisheries, and that's true everywhere mangroves grow. There is actually Nipa frutescens, right? The mangrove palm. It's a really incredible plant, and it's one that people have used for both biofuels and for some forms of palm wine. And I think I've even read that the fruits are edible and pretty good. So that would be in places of the world where mangrove palms grow, a direct way that people can actually eat from mangroves. But I don't know of any direct nutrition that happens for the mangroves that grow in the New World. So yeah, it's pretty much all through the fish.
And eventually, are you going to publish your paper, your research you've done on here? Somebody asked if you have a copy of your presentation, which, well, there's a copy of this presentation, but I think he meant, or are you publishing your results anywhere in the future?
That is absolutely my goal. Okay, so the first paper from this project is really close to being published, and that's the one that presents the tools that we built to figure out how big the invasive trees are from simple measurements. So anybody who's interested in trying to figure out how much carbon is in that carrot wood that's in the common area behind your house, right? Send me an email. I'll send you the paper. You can measure its diameter, and you can figure out how big that plant is. So that one is just about ready to go to a journal for publication. There are two other papers that we're currently working on. One describes the kind of vegetation results at Tidy Island. Basically, the second result of seeing how all these invasive trees interact with one another and the leaf litter, and the results of our experiments, both in the lab and in the field, to see how native plant diversity responded afterwards. So I've completed all those analyses. I've got some of it drafted. It's not quite all put together yet. The final one will be the one that presents the results of the carbon. And that one is really just hot off the presses. Like, I completed these analyses maybe 3 weeks ago, but for anybody who doesn't have access to or is not super interested in the primary scientific literature, that's basically like papers where I'll be publishing these results, we are putting together a restoration guide that will be made available through the Sarasota Bay Estuary Program's website. And that target audience is going to be practitioners, right? So people who are managing these kinds of habitats in Florida and elsewhere. And the tone there is going to be different, right? It's not going to be about hypotheses and statistics. It's going to be, what do you do when you've got these invasive plants and you want to promote native plant biodiversity, you want to promote carbon sequestration, or you want to promote fish habitat? Our objective with that restoration guide is to map out some of these trade-offs in quantitative terms that will help people prioritize what kinds of interventions to take depending on what kinds of habitats and services they want to see. So stay tuned for that. We've got to have it done by May. So that will be the very latest that is put together.
The seedlings you mentioned that came up after the mulch was removed, did those just, they were there basically, or they're all naturally came in, the native plants, seedlings?
Yes, that's a super good question. Most of them were spontaneously recruited, right? There were a few of our plots where we caught trees that we intentionally planted onto Tidy Island, but those actually didn't drive the results that I'm talking about, because those were mostly in slightly different places. They were mostly plants that either, I had seen growing at Tidy Island, just they were uncommon, right? And they became common enough after we controlled the invasive species for them to show up in our inventory, or they were completely absent from Tidy Island. And it took removing the invasive trees in order to give them the opportunity to get established. Mm-hmm. So an example of one that I did not see at Tidy Island before we did our restoration project was trumpet creeper, right? So Campsis radicans, super nice plant. And it could have been growing at Tidy Island, but I had marched all over that place and I had never seen a trumpet creeper. And then within a year of controlling the invasive plants, one of them just showed up there, right? And, not super surprising. I think those things are bird dispersed. But it is pretty impressive that it just spontaneously recruited out there over that span of time. There were a few other kind of cool finds that we saw in the native plant community, but a lot of those plants were pretty little. And so it's hard to know if they're going to stick around for very long. And they're not generally things besides trumpet creeper that, Tom, you would want to sell to people. We're talking about things like rustweed, like polyprimum procumbens and stuff like that. So, fun if you're crawling around on your belly with a hand lens, but not exactly the kind of thing that you want to stick in your garden.
Just a couple other comments. Horrible that invasive removal is so carbon expensive. Really fabulous presentation. Nipa frutescens produces, I guess that's the other mangrove, the fourth mangrove, produces sugar, vinegar, and alcohol from the flower stalk. And the young seed is edible. So one of the mangroves is edible, not the one that grows here though, I guess.
Yeah, that's that mangrove palm. Super, super cool plant.
Mangrove palm.
Yeah, it has a really long fossil history. There are fossils of mangrove palm in sediments in London, right? So even though it's restricted to Indo-Malaysian mangroves today, it was around a lot more extensively before. Probably be a bad idea to try to grow it in Florida though, because I guarantee that thing would get out. That is a very, very tough plant.
One thing I find interesting is that the invasives store so much carbon, and by taking them out, you're taking away all that carbon storage there, which seems kind of counterintuitive to getting it back to the natives. I'm trying to wrap my head around that.
It's definitely not the story that I would like to share, right? I would like to share a story that you can have your cake and eat it too, right? You can get rid of these plants we don't like, promote the plants that we do like, and do it in a way that is more climate friendly. And for reasons that, I was hinting at in my discussion with Karen, it is possible, right? 10 or 20 years from now, we might see so much carbon that shows up in the mangrove soils. And so much carbon that is in this more diverse community of native plants growing on Tidy Island that it does kind of balance out. But that's a long time, and it's a deep hole, right? We lost a lot of carbon from getting rid of those invasive plants. And, you know, in some sense, it is, I think, not too shocking because those plants are just so big and they grow so fast, right? Like part of the reason why they're so competitive is simply that they are so good at storing carbon. They just put it in their wood and they use the carbon in their wood and in their leaves to keep any other plants from trying to grow. And for that reason, I feel like what we saw at Tidy Island could be a story that would show up in lots of other habitats where invasive trees are really a problem, right? And it's probably going to wind up being some hard choices that habitat managers are going to face. Do you get rid of these invasive trees that store a lot of carbon, right? Or do you have more diverse communities of more desirable plant species, even if it means you're not storing as much carbon in those habitats?
Yeah, Julie mentioned that Australian pine is the leading carbon sequestration plant in urban Miami. There's a lot of Australian pines in Miami down there. So Elliot wrote, is there any plan to remove more invasives out on Tidy Island? And since your project's wrapping up, I don't know what the answer to that would be, but.
Yeah, so quick comment in response to Julie's, which is I think a good one, right? The best way in my experience of dealing with Australian pine is to turn it into furniture. That way you're not losing the carbon in the atmosphere. It's gorgeous wood. It is super hard. Like I've got this cutting board on my countertop up here in Providence that is actually from one of those trees that came down on the Ringling Causeway during Hurricane Irma, right? There was a local artisan who went out, he salvaged that wood and he turned it into cutting boards. It was like a really wonderful thing to take away with me. So if that's what you're doing, like if we can actually turn it into timber, it doesn't wind up back in the atmosphere. And so some of the like timber salvage shops in the Bradenton area up along 301, you can actually get a really nice tabletop, right? That's a live edge slab of Australian pine that presumably came from some kind of a place where it was removed. So that is what I would advocate doing with that plant, right? Keep it out of the atmosphere, put it in our homes. I think that's a really nice way of dealing with it. In terms of additional invasive plant management at Tidy Island, you know, I was in conversation with the people in the condo association. They've been really excellent neighbors, and they have quite a bit of property that is actually overrun with invasive plants there. And they were interested in applying some of the best practices that we learned about through our experiment to their own property. So I think that would be a really cool outcome. And we'll see if the people there are still interested in doing it. As far as the parts of Tidy Island that are part of the new College of Florida Foundation, I mean, it depends on a couple of things, right? Like we controlled most of them as part of this project through the Environmental Protection Agency. There are still a couple of areas that we didn't touch, basically because we didn't have the money and they weren't part of our experiment. So there's an opportunity there to control those plants. And then there are these little tiny spoil mounds scattered throughout Tidy Island that are dredge spoil left over from the mosquito ditch dredging. And those would be really tough to get to because they're so far back in the swamp. I think that any effort to control invasive plants in those places would be pretty expensive and probably wouldn't have a lot of return on investment in terms of habitat actually restored. So, you know, the opportunities to continue to do this invasive plant control at Tidy Island will depend on resources, right? Can we find another grant to do it? And then also the priorities from the new College of Florida Foundation, right? If they prioritize investing in this property and the deeded mission and purpose of Tidy Island, which is for research, teaching, and stewardship, then I think there's an opportunity to continue to do this kind of habitat management. But fundamentally, it will depend on people in the new College of Florida community, right? What kinds of goals they set for this habitat and what kinds of resources they can put into it. And I'm hopeful, right, that I'll be able to continue to stay engaged in those conversations so that this really incredible resource that we have, Tidy Island, can continue to change for the better, the way that it has over the course of the past 5 years or so since we've been working with people in the community on this project. Was that a nice diplomatic response? I hope so, because that's actually the outcome that I would love to see. It's just going to take sustained effort to get that outcome to happen.
Well, we got lots of thank yous. Thank you for your presentation from Andy. All your hard work in the swamp that went into collecting all the data. Interested in your recommendations to land managers. Very selfish reasons. I'm a land manager at Crystal River. So thank you for the presentation, Brad. I've learned a lot. Let's keep all those native babies planet thrive and make up for the carbon absorption. Thank you. Fascinating. Sean wrote too, there's a bio blitz too, that's off the subject this Friday that they're going to doing more eco hikes by Selby Gardens coming up next year. So keep your eye out for that. And there's a bio blitz this Friday. If you're interested, it's in the chats. You can Google that and find out more information on that. So anybody have any other questions for Brad?
Well, regarding the BioBlitz, is that a private chat? Because I don't see it.
It was way up in there. I don't think it was.
Oh, further up.
It's
Okay.
further up there. It's way up. Yeah, to everyone. No, it's to everyone.
Okay.
It's about the fourth one down. So
You can find the information on the Selby Gardens website, and it happens to be this Friday at Tom Bennett Park.
Tom Bennett Park. Well, it was very interesting. I learned a lot too, Brad. It was a very nice talk.
Well, thanks a lot. Just generally for people showing up for the presentation, Tom, Allison, other organizers for inviting me. It was really cool to come back, like 8 years after or 7 years after my first presentation to the chapter. I also specifically want to give a shout out to Karen and Julie and Jono and Lauren, actually, who all came out to Tidy Island and helped put plants in the ground at different points in time. So, I was out there in January to get all of our equipment out, and it was such a gratifying feeling to see how that place has changed for the better through effort on the part of so many people in the community. And it was really, really nice thing. And I'll be back for graduation and a couple of other things a little bit later on. So hopefully we will cross paths the next time I'm down in Sarasota.
Hopefully some of the students that were involved in the project will kind of monitor it on their own, possibly, and try to keep out some of those carrot woods.
It's a tough fight, and yeah, we'll see how well we do.
Good. Well, I think we're going to keep the page open. The meeting's pretty much over, but we were going to keep the Zoom on if anybody wants to just say hello to other members or sign off or have a good evening. But thank you all for showing up this evening. So field trip will be on Saturday and should be a really good one also. They've done a lot of work out there at Lefus Key, and there's lots of mangroves to see up there.
All right. Great to see you, Brad. Thanks.
Karen, good to see you too. Thank you.
Yeah.
Bye.
Thanks, everybody. All right.
I hope we don't wait 8 years to get you back, Brad. That was really a great presentation.
Mhm.
Yeah, it was great.
Yeah,
Thanks.
I mean, yeah, I like doing work in the community, and this is a complicated project. So it took a long time to get results that we're sharing. But they're cool. And I really stand by them, I think.
But part of the interesting part is watching how you develop it and figure it out. So, I wouldn't, if you come up with another project in Florida that might be of interest to us, even before you get results, maybe you can let us know that you could be a speaker again.
Yeah, no, I'd gladly do it. Yeah, Tidy Island, there are opportunities to keep doing work there, so there could be an update down the road. And then the other project that I still have going on in Florida is actually looking at Tillandsias. So I've been working with folks at Selby and some people actually at Fairchild Tropical Botanical Gardens in the Campon, learning more about the weevil and what kinds of impacts it has on the local plants. So yeah, those projects are starting to yield some fun results. So that'd be fun to talk about at some point in the future too.
Yeah, that would be an interesting topic, actually, the bromeliads. So our Tillandsias, there's so many of them, so many people are familiar with one of them or two of them. That's it, but there's quite a few species of Tillandsias.
Yeah, I missed out on a walk through the Fakahatchee back in January, where I think they tallied 9, which is pretty cool. So yeah, they got the Guzmanias and a couple of the other like really fun things that are just way back there in the swamp. I've seen them before, but it would have been nice to see them again.
Yeah, that's always, Mike Owens is always a fun trip leader down there. So and it might have been one of his last trips that he's leading, because I think he's left the Park Service now. So more he'll do it on a volunteer basis, I think, at this point. So
Awesome.
Andy, where are you at Crystal River Preserve?
Yes, sir.
Nice.
We
Lots
might
of
need to visit you sometime.
Sounds good. Yeah.
So how did you hear about our presentation for Serenoa from up there?
One of my, he's actually in the call right now, Trent Berry.
Oh, Trent, Trent.
Yeah. He was
That's
one of
right,
our
he's-
AmeriCorps last year. So he let me
Good.
know what's happening. There's a lot of overlap here between Tidy Island and some of our property, with archaeological remains and things like that as well. So yeah, it all ties in. So yeah, I'm very interested in
Great.
learning more and seeing this get peer reviewed and maybe taking some lessons home.
Sure.
Awesome.
Well, Trent,
Yeah.
thank you for getting people coming to our meeting.
Yeah, for sure. Yeah, and Andy, you saw my email there. Just drop me a message and I'll make sure that I keep you informed of our results as they come out. And I'd be glad to send you a copy of the presentation and a couple of the other materials I've already put together. All right. And then actually a question for Allison and Tom. Actually was trying to get my program officer at EPA to jump on the call, but he was traveling. Could one of you point me to where the recording is? Because he specifically asked me if there was going to be a recording, if I could share it with him.
Yeah, we uploaded it to our YouTube channel for and all of our prior ones have been recorded are up there too. But if you want, I have your email, and when I finish the link, I'll send it to you. So you don't even have to go up to YouTube if you don't want to. You can just have the link.
Allison, that's fantastic. That'd be perfect, because I can just forward that right to Troy at EPA. That'd be great.
Sure. And if it's too big of a link, then he'll want to watch it at YouTube, and I'll just, not the link, but the whole recording. When I get the recording, if I can send it and it's not too big, I'll send it to you. Otherwise, I'll send you the link.
Perfect. Thank you so much.
Sure.
Good deal. Well, like I said, next September, we're going to start meeting at Selby again. So we'll be back to in person for those who choose to, but you can Zoom on it also. So we're hopefully everybody won't Zoom and some people will show up in person, but that's to be seen. So, well,
but, with the wonderful plant raffle that you always have for us, I think that'll encourage people to show up.
Yeah, that's a great reason to show up.
Yeah.
You have to have donuts, we'll have donuts. I mean, whatever.
We have things of some other incentives to get people to show up in person. Definitely. So we're good.
Awesome.
Yeah, well, this is great. Thank you, Brad.
Thanks a lot, Brad. Okay.
Yeah, my pleasure.
Bye, everybody.
Close it off. Thanks everybody for coming. Trent, take care. Chris, see you soon. Okay, everybody. Thanks, Allison.
I'm gonna hit the button and end the meeting.
Okay. All right.
Bye, everyone.
Bye-bye.